at Harvard have developed this extraordinary video that really talks about the dynamics that are going on all the time as these blood cells flow through the system and ultimately bind through the intercellular communication to the cell surface of the intimal tissue layers and transfer their information deep within the cell through this membrane connection. The membrane connection is built on the lipid bilayer of the cell. As you recall, the lipid bilayer is made out of these cholesterol rafts that have polyunsaturated fatty acids in them that are altered and modified in their function by diet. It transfers then its information through this lipid bilayer, through the transmembrane protein binding sites, ultimately down through the deep actin, myosin fibrils that make up the internal skeleton of the cell. The skeleton is not just a bunch of cellular soup washing around like a baggie, but actually it's a very gelatinous material that's three-dimensional and structured and functional in its own representation. Within the cell, you have the assembly and deassembly of these transport molecules and these fibrils that are the guide paths and the pathways that will ultimately lead this information from outside the cell to the interior of the cell, which I call the brain of the cell, which is the cell's brain, which is the genome. Ultimately, this transferred information through replication and deassembly, assembly and deassembly, then allows for the message to pull materials like these macrophage aligned materials that are ultimately on the organelles into the internal interstices of the cell, where they're going to stimulate specific organelle function, like mitochondria, ribosomes, Golgi apparatus. As that happens, you get this nuclear envelope, which then is going to penetrate, the information will penetrate and turn on specific gene response reporter regions. These genes then get lit up and they start to make their own messenger RNA. As that messenger RNA is made, it's the template to make certain proteins. Those proteins then travel to the organelle specifically to activate and turn on function, like the energy powerhouse of the cell, the mitochondria. These ribosomes then completely produce individual proteins in response to the environmental stimuli, which was triggered by the attachment of the white cell to the vascular endothelium. As that occurs, you get recruitment of various types of internal structures of the cell that bring new information to then process this new environment that the cell is experiencing on the surface of the endothelium. That then relates to the release of various proteins. These proteins get secreted both internally to the cell and externally through membrane binding ports. They then transmit information extracellularly that then go into systemic circulation and say, stuff's happening, guys, be aware of this. Those information molecules that are being produced within the cell on this transmuted process through its transcription, translation, and ultimately excretion controls then the regulation of how that white cell will ultimately extravasate, travel in through the vascular endothelium, and now change its form and function into a foam cell to become an atherogenic endocyte. There's the connection in